Thanks so much, Sam. So for those of you who are new to this meeting, we have a tradition of holding questions for the break because we want to pack in the science. So the speakers will be around, so just write your questions down and don't forget them. So the next speaker is Dr. Ralph Weissleder. Dr. Weissleder is the Thrall Professor of Radiology and Professor of Systems Biology at HMS and the Director for the Center for Systems Biology at MGH. He's also an attending clinician in interventional radiology at MGH and a member of the Department of Systems Biology at HMS and the Harvard Cancer Center. He's also a member of the National Academy of Medicine and the American Academy of Arts and Sciences, among many other appointments. He, together with Bob Langer, actually started our Center for Cancer and Nanotechnology Excellence here at MIT, which laid the groundwork now for our Marble Center. The title of his talk is Developing Next Generation Diagnostics for Cancer. Welcome, Ralph.
Perfect. Good morning, and thank you so much for the invitation. It is a pleasure to be here and share with you some of the exciting developments in translational diagnostics. So in the era of precision oncology, we rely on three diagnostic pillars to provide deeper and longitudinal analyses. And these three pillars include, first of all, imaging, as we just heard, at the anatomic, physiologic, and molecular scale. They include tumor biopsies, be it through surgery, through image-guided needle placement and removal of core tissues, or fine needle aspirates through skinny needles, and finally, liquid biopsies, where we sample peripheral blood to interrogate that blood for circulating tumor cells, circulating free DNA, or extracellular vesicles. Now, despite huge advances on the research side over the last decade, there remain clinical challenges and problems. And so it's one thing to do something in the lab, and the other one to do sophisticated diagnosis every single day in the clinic. And so from a clinical perspective, the needs are as follows. First of all, we need to improve existing diagnostic tools with much faster turnaround time. You might be surprised, but when we send biopsies for sequencing or pathologic turnaround time, these turnaround times can be anywhere between 1 week and a month. And so clearly way too long, and the technology is there to do this much faster. To allow more frequent sampling in the same patient, lower morbidity, developing technologies with higher patient acceptance and higher accuracy, focusing on diagnostics that have actually actionable consequences, i.e., whether a patient will benefit from an expensive treatment or to prevent toxicity and side effects, enable earlier diagnosis, as we just heard, and finally, technologies for improved data visualization to take heat maps and this massive amount of information and link it back to actionable things to do in the clinic. And all of this, of course, at affordable costs. And so many of these challenges are probably straightforward, and we think, well, they're logical, but there are real bottlenecks in the clinic. So, of course, what's needed are interdisciplinary teams to build the right tools for the right problem. And so what I'll do today in the next 20 minutes, 25 minutes or so is go over three different vignettes of new technologies that we have developed in our laboratory. The first one is rapid cellular profiling technologies of near single cells obtained by fine needle aspirates, a technology which we call scanned single cell analysis of tumor cells. The second one is using nano-plasmonics to profile circulating tumor vesicles in plasma. And the third one is technology, inexpensive technology for global health and point-of-care diagnostics to bring molecular diagnosis to low and middle income countries using artificial intelligence diffraction analysis. So let's start with the first story. So fine needle aspirates, as opposed to core biopsies, are done with very small needles on the order of 22 gauge needles that are about 0.5 millimeter in size. So these fine needles have complication rates that are approximately 5 to 10-fold lower than core biopsies. And one obtains cells like these here that are then stained, analyzed by cytopathologists with conventional chromogenic stains. The cells on the left side of the screen here are malignant. These cells here are benign. And you can already see that we get into a gray zone in between where it is hard to differentiate malignant from benign tissue. And this is sort of current state of the art. So the other challenge here is that the number of cells that are harvested with each pass is fairly small, on the order of thousands of cells, so not millions or billions. There are host cells in there in addition to tumor cells. There's usually no embedding, so these cells are basically attached on the glass surface. And there's a limited number of cytologic stains. And so, for example, if we were interested to measure mutant proteins in this cell here to really figure out whether it's a malignant cell, it would not be possible because the unstaining of the stain is a problem and the cell would probably get washed off. So we have been very interested in developing methods of deeper protein profiling in cell aspirates such as these. And so the method that we currently employ uses monoclonal antibodies that are DNA barcoded. And this DNA barcode has a complementary strand with dual fluorochromes on it. And so it turns out that through the judicious use of a melting buffer, a low salt condition melting buffer, this fluorochrome can be simply washed off. We then cap this strand with a non-fluorogenic DNA to prevent the reassociation of this fluorochrome. And so this process can be repeated several times using different antibodies and different fluorochromes. So in the clinic, it would look like this. Lesions are sampled. We come in with a set 1 of monoclonal antibodies, anywhere between 4 to 6 colors with this strain. Wash this off, come in with a set 2, and so on, and cycle through a couple of times. After each incubation, image these cells, and then use artificial intelligence and deep learning algorithm where the computer basically analyzes these cells and puts everything together. As you can see from the timeline down there, we're down to several hours. So these are tests that can be done within the same day in the clinic. So taking this into clinical trials, these are the key pathways that we were very interested in profiling. In other words, patients who were being enrolled for PI3 kinase inhibitor treatments or CD4K inhibitor treatments. The question was, what happens to the protein expression levels all the way from up here to down here in single cells in our patients? And so in order to do all of this, we did 2 validation studies. In the first set of about 10 patients, we asked a simple question: what are the protein levels in different patients? And so each column here is a different patient. This is a scaled heat map. And first of all, you can see the protein levels are all over the map. This patient 16 here has much higher P10 level as other patients. And we said, okay, it is what it is. It was not unexpected that this was the case. But what was clear was that the phosphoproteins, for example, phospho-EBP1 over EBP1 to phosphoproteins were always lower, and we knew that that was going to be the case. And then we said, well, what if we express these on a per per patient as ratios. So for example, phosphorus 6 over S6. And the second experiment that we did was dose response, where we measured this in patient cell lines and in patients with different doses of drugs such as this P110 inhibitor, and we can see beautiful dose response curves, both in cell lines, but also in our patients. And so taking this further, I'm going to show you a couple of patients that were biopsied repeatedly, and repeatedly up to 3-4 times during a 2-3 week treatment period. So in this particular example, we're looking at about 150 or 200 cells that were obtained with a single FNA, looking at the phosphorus 6 over S6 ratio, wide distribution. As expected, upon treatment, this is about a week treatment of P110 inhibitor and CD4K inhibitor, this spread decreases and the overall mean also decreases. And we found similar things of all the patients who responded to those treatments, a decrease and flattening basically of the heterogeneity. Not only for this particular ratio, but also for others such as phospho-AKT over AKT and EBP1 and so on. In this graph, one of these patients did not respond and clinically, and we were able to basically show that this was predicted through serial biopsies. So at the end of the day, we now have a tool in hand to do basically pathway analysis, same-day pathway analysis as patients come onto molecularly targeted trials. The second example I want to show you, or set of technologies I want to show you, is to analyze these small vesicles that get shed by cancer cells. These vesicles, extracellular vesicles, have unique characteristics. They're extremely abundant in the bloodstream at the level of 10 to the 9th to 10 to the 11th vesicles per milliliter of plasma. They're extremely stable, as opposed to circulating cancer cells, readily accessible. They contain protein and mRNA that is reflective of the cell of origin. So from a clinical perspective, to be useful, the key challenges and questions are, with a blood draw, how do we separate vesicles that originated from the cancer versus from host cells? How do we keep them apart? Which EV should we best look at? Is it exosomes? Is it microvesicles? Or is it other fractions? And lastly, what kind of technology should we use to keep cost downs and increase speed? And so over years, and this has been a little bit of trial and error, our lab has developed a lot of different technologies for the isolation of these vesicles, for protein profiling, and for mRNA detection. And I won't go into all of these technologies as this has just been reviewed. I'll bite to say that two technologies, nanoplex using plasmonics and electrochemical sensing, are making their way into the clinic. And perhaps the furthest advanced is this technology here. And so this works as follows. It's basically a surface plasmon resonance technology, but not in reflection mode, but in transition mode. So on glass slides, we deposit gold films that have periodic holes. And we have these periodic holes so we can shine light through the gold film. These gold films are modified with monoclonal antibodies around these holes to capture EVs of interest. And whenever these EV bind, and green light is shown through these nanoholes, by virtue of binding, there is a spectral shift of this light, and this is the spectral shift that we measure. The degree of spectral shift correlates with the amount of binding. So this technology was first tested in ovarian cancer and subsequently in pancreatic cancer. And the first question that we asked was what kind of biomarkers to go after, or what kind of proteins to go after. And we tested about 100 different proteins and finally settled on the analysis of the ones shown here, the typical ones. EpCAM, GPC1 went to GRP94, and so on. And so in these 25 patients, first patients, where we drew blood and asked what kind of proteins do the vesicles express, it becomes clear that there was not a single protein that uniquely identified all patients with pancreatic cancer. However, when we summed some of these up here, 5 signatures, and derived this, what we call PDAC, extracellular vesicle signature, most of them did show up. Interestingly, compared to pancreatitis or to patients who had other things happen in the abdomen, were operated for that, this was fairly negative. We subsequently went on and tested this in around 150 patients, and the diagnostic accuracy as it stands right now is somewhere in the 90% and with a specificity of 85% from a simple blood test. So where do we go from here? We have reason to believe that we can make this much better. So first of all, choice of biomarkers. So not only for pancreatic cancer, but also for GBMs, colorectal cancer, ovarian cancer, breast cancer, lung cancer, and so on. There are proteins that are more indicative of whether we are dealing with cancer than others. And so we understand this whole world map of which biomarker is most suited for the detection not that well. So much more work is needed, starting with proteomic analyses of patient samples, primary patient samples. The second thing that needs to happen is, as these vesicles are shed from dividing cancer cells, it became clear that the vesicles are not homogeneous. They're actually their protein makeup is actually fairly heterogeneous. We discovered this by chance by developing imaging methods to image and profile single vesicles. And when we do this and look for different proteins in vesicles, this is sort of the landscape here. This is one example of microvesicles obtained from a GBM neurosphere that was analyzed for these proteins here, and green is high and blue is low. And as you can see, a previously thought ubiquitous marker of these microvesicles, integrin beta 1, was only present in about half of the vesicles, and the other half were absolutely negative. So it is very important to better understand the makeup of these vesicles to define which markers are best suited and are more indicative of disease. And so this is work that is currently going on. So the final vignette and story I wanted to share with you is technologies that we have developed for remote diagnostics in Africa, where there is no access or very little access to complex labs. We believe that molecular diagnostics are possible in these countries, and the lab has been working on approaches to make this happen. So in other words, particularly in sub-Saharan Africa, does this patient have AIDS, does this patient have TB, or does this patient have lymphoma? And if lymphoma, is it really aggressive, or is it an indolent lymphoma? And just putting things into these 4 buckets would make a huge, huge difference. So we developed technology for the molecular analysis of these mass lesions. So this started in 2005 when we had shown, and many others had shown, that smartphones could be used for molecular diagnostics. And so smartphones, so while people have made optics to convert them basically to mini or cheap microscopes, we reasoned that that is not the way to go, because if we just have a cheap microscope, we still need a pathologist, and the big problem in many low and middle income countries is the lack of pathologists. So what we developed was somewhat different. We used nanobeads to decorate cancer cells, so immunobeads to decorate cancer cells, and recorded holographic signatures that these nanobeads emit and reconstruct these diffraction patterns. So, so this worked great. It was published a couple of years ago, and then as we were trying to roll this out, one of the biggest problems was that the smartphone technology was changing on a yearly basis, was way too fast for us. So we decided to build our own box, basically, that still has similar dimensions. It's 10 by 15 by 10 centimeters. But the imaging module has a much larger field of view, so we can image many more cells compared to a smartphone. It has improved spatial resolution, enhanced computational power. There's a Raspberry Pi on the inside. And it uses deep learning and multiplexing to do all of the cellular analysis on the spot in the machine, and it costs $180 to make. So it's priced for low and middle income countries. There are big things under the hood in that the algorithm that looks at these holograms, first of all, identifies whether we're looking at cells, whether we're looking at ghosts, or whether we're looking at dust in there through cell recognition. It then, for each cell, goes in and asks, what is the expression level of a given protein of interest? And we do this for many different proteins. And then it goes into a decision tree algorithm to define what kind of underlying problem there is. So this is an automated analysis of many of these cells on a typical field of view. And at the end of the day, this technology is as good as flow cytometry. So $180 technology is as good as a $200,000 flow cytometer for very rapid analysis of fine needle aspirates. So again, we took this into a clinical trial at Mass General where we enrolled 40 patients who had lymphadenopathy or enlarged lymph nodes somewhere in the mostly abdominal and periaortic area. These were all needled and aspirated. We took one aspirate for this holography device, and the rest of the samples were submitted for pathology. I'm going to skip over this, albeit to say that about 24 of these patients were lymphoma patients and the rest did not have lymphoma. The overall diagnostic accuracy of this test, so the question, does this patient have lymphoma or no lymphoma? The sensitivity was 91% with this method, the specificity 100%, and it was higher than conventional flow cytometry, interestingly. When we asked the question with different markers, does this patient have aggressive lymphoma versus indolent lymphoma, so different treatment schemes for these two, the sensitivity was 100% and the specificity around 80%. So this is currently being rolled out actually into the same hospital that Sangeeta mentioned at the beginning. This is going to Botswana, to Princess Marina Hospital, where we got funded to study 400 patients with a suspicion of lymphoma. And so it's totally, there's an app for this as well. It's totally automated diagnosis. So a healthcare worker would stick a needle into one of these lesions, squirt the sample into a little plastic cassette, and everything else is automated at $180 per device. So I'll stop here and recap a few things. So overarching themes of our diagnostic work are to go from tissue sampling to single cell or scant cell sampling, simply because these interventions are much less invasive, much better tolerated by our patients, and way fewer complications. From conventional staining, we're going to quantitative multiplexed pathway analysis that gives us more information. The clinical priorities are clear. It was mentioned several times: early detection and treatment informing diagnostics. Miniaturization and nanotechnology allows us to make these devices much more affordable than they have been in the past, with implications for global health, but also for screening in our and repeat testing in our clinics here. And the last point that I did not go into, didn't have time, is the whole concept of visualization. How do we actually visualize the data in the electronic age and electronic medical records, so it becomes interpretable. So with that, I'd like to thank people in my lab who have contributed to this work, as well as the many, many MIT students who helped us build the last device, as well as the many collaborators locally and across the US and internationally. Thank you very much.